I've been involved in rare diseases and gene therapy for 4 decades, spent most of my time in the academy, but because of the concerns as to system failure, we left Penn a year and a half ago and started a benefit corporation called Rare Therapeutics, Inc., which is a for-profit but has the mandate to also address the needs of patients and populations dealing with rare diseases. So I want to tell you about our strategy today and what we think could be deployed to make this a successful enterprise. Success meaning it's sustainable. First, best-in-class technology. That's what we've done for most of the time that I've been a scientist, and we'll share with you a few of our platforms. The second is to focus on unmet need, which is basically most rare diseases and patients living with them. The third is therapeutic platforms, and Lawrence brought this up at the plenary talk, and really today what I'm going to share with you is the idea of platforms, what they are, and what advantages there will be in developing products as platforms. We need a different commercial model, and for rare diseases we believe that centers of excellence in a hub and spoke model is the way to go. And then innovative partnerships, such as the one that we forged with the Ministry of Health in Brazil and also the Department of Health in Abu Dhabi, where governments that are responsible for providing health care are aligned with what we want to accomplish. So I'm going to share with you 3 different platforms based on genetic medicines, and 2 of which we'll illustrate with data. The first is our AAV-CNS gene replacement therapy platform using AAV9-like vectors. What we showed in non-human primates is that we can accomplish efficient and durable expression in the spinal cord, less so in the brain, but still pretty substantial, focusing for global neuropathic diseases on lysosomal storage diseases, and for spinal cord diseases, sensory motor neuropathies. We'll talk about the LSDs. We also developed an engineered capsid that has a higher tropism in the heart and the muscle, and focusing on monogenic diseases that affect myopathies of the heart and the muscle. I'm not going to share with you those studies. We're going to present our work at the American Society of Gene Therapy on Wednesday. And then finally, liver metabolic diseases, a genome editing play that we initiated in 2015, and another company I started, Eacure, that has a UK story that I'm delighted to share with you at the end. This is where we are looking for targeted insertion of a gene in a dividing liver, and this shows in non-human primates with the donor vector as well as the nuclease, we can achieve levels of integration at 22%, between 20 and 30%. So the platforms, the CNS and the liver platform, I'll share with you. We have clinical data in those and what we've learned about how powerful they are and what the limitations are. So for the idea of a platform in any genetic medicine is, I think, best illustrated in our CNS platform, where we have a delivery vehicle, we have a method of production, we have a route of administration, we have a dose, and we have a gene that treats a disease. And if it's a platform as we develop it, all we change is the gene, keep everything else the same, different disease, change the gene, different disease. And the hypothesis is if you keep everything else the same, that could be very useful in informing the subsequent products that are developed under the platform. So what we've done in this platform is evaluated this in over 570 non-human primates across multiple programs, 8 clinical trials that are underway using this platform, in which we deliver the vector into the cerebrospinal fluid. So what we've learned, sort of high level, is that there is strong read-through in terms of preclinical safety and pharmacology into the human studies, as well as molecular and biomarker correction. So what we learned in the first disease, we've seen throughout the other diseases in the program. that the manufacturing processes and the attributes of the product are shared, so there's some economy of scale. Where we have disease-specific challenges are human natural history data, real-world data, that will be disease-specific. And the actual translation to clinical benefit has a component of the disease that will affect that. The reason that we've selected as our early candidates for this platform the lysosomal storage diseases are that limited gene transfer, 5%, 10% of cells in a globally diseased brain has the potential to treat all of the neurons because of what's called the bystander effect, that the gene expresses an enzyme that's secreted and cross-corrects. That there is the potential in these diseases to qualify for what we in the United States refer to as accelerated approval, and that is an approval that is based on biomarkers and natural history as comparators, because they're serious diseases and there are biomarkers in all of these diseases that are believed to be related to pathogenesis, and they're basically whenever the substrate accumulates because of a defective enzyme, that the accumulation of these substrates can be used as biomarkers, that when you correct that you are correcting at least the biochemical and the genetic and the molecular aspect of the disease. I started a company called REGENXBIO that has brought 2 programs forward, one for MPS I, MPS II, Hunter and Hurler disease. And then the 3 programs that we have brought into rare TX, the leukodystrophies called GM1, leukodystrophy, Krabbe, and MLD. And we have, all of these are clinical stage programs. And I'll just share with you the story with GM1 gangliosidosis, the one that's the most advanced of the gangliosides. But this is a defect in a gene called beta-galactosidase that, when defective, there is an accumulation of substrate called GM1 ganglioside. And this is believed to be involved in pathogenesis of the disease. As far as the clinical phenotypes, it's very severe progressive neurodegeneration with death over really only a few years of life. And the disease is stratified in terms of the most severe form, the early infantile, and attenuated forms of the disease. So we, at a company, Passage Bio, that I had started, a trial. We've enrolled 11 subjects. When that company basically hit the wall, literally, and is now being dissolved, I think this week or next week, we salvaged those programs by licensing those from Passage Bio into Rare TX, and now we're in control of those programs. The Imagine trial is a phase 1/2 open label study, and it would be phase 1, 2, and 3 if the results are encouraging. Assess safety and tolerability, and it's a single dose in early infantile and late infantile patients. This is a multi-center trial, and out of the 11 subjects, I think 3 actually were enrolled here in the UK at GOSH. And I'll just share with you, because we have a lot of data, about the middle and the high dose of the late infantile patients, looking for evidence that we have biochemical correction that's sustainable. So this shows in 3 subjects the beta-gal and the cerebral spinal fluid, which at baseline is here, low, and this is normal. And in all of them, it goes actually above normal, and for the times that we followed them, is stable. So one-time administration has the efficiency and the durability. Then in terms of accumulated substrate, this is what we saw in terms of the elevation of GM1 in the CSF. This is normal. And you can see following gene transfer, all of those are trending to and eventually achieve normal levels. So we're now in the process of evaluating the data and having discussions with health authorities about next steps. So the second platform is based on a liver metabolic disease that is called ornithine transcarbamylase deficiency. So this is a defect in a gene called encoding the protein OTC that's part of the urea cycle. The importance of the urea cycle is that it catabolizes ammonia, which is toxic to urea. And absent that, patients are susceptible to hyperammonemic crisis whenever there's a protein overload. This is X-linked recessive, so little boys generally are more severely affected. And we've gotten to know a family called the De Leon family that illustrates the spectrum of this disease. The neonatal onset form of the disease, babies are born normally, but then they're noted to be sensitive to protein, and in the acute neonatal period can develop a lethal hyperammonemic crisis. They could be maintained on medicines, but they need to be listed for a transplant, a liver transplant, that is a form of gene therapy. Otherwise, they'll succumb to repeated hyperammonemic crises. And this is her second son. The first one died of OTC deficiency at 3 days of age, and this is Michael, who actually was transplanted at 7 weeks of age, which is remarkable, but died of complications of liver transplant at 7 months. Now, heterozygotes can also manifest with symptoms, and this is Ariel, her daughter here, who was diagnosed at 7 months of age, but they are at risk of repeated hyperammonemic crisis, and they can be maintained, and these can largely be prevented by very aggressive protein restriction and substrate reduction therapy. So the challenge of treating a newborn versus an adult with gene therapy using an AAV approach is that the target cell, the hepatocytes in newborns, are dividing, and AAV doesn't integrate into the chromosome. But if it delivers its gene in a postmitotic cell, like a heart, a retina, a neuron, possibly skeletal muscle, it'll be stable. So what we would expect to see if we treated a newborn with an AAV gene therapy approach is we get a number of cells corrected, but as the liver grows, these are diluted out and you lose efficacy. So you really need a therapy in which you can integrate the vector genome into the chromosome, and that's genome editing, in which integration, if it does occur, then as those cells divide and as the organ grows, you maintain the same efficiency of gene transfer and expression. So we developed an approach in 2015, right, a little bit after CRISPR was identified or described, using a targeted integrase. So what we did is, in collaboration with a company called Precision, I started a company called Eacure, where we developed a nuclease that would create a double-stranded break in a safe harbor. It turned out we used the PCSK9 locus, and then introduced the donor gene to integrate into this specific locus, then expressing human OTC. This was a 2-vector AAV strategy, one expressing the nuclease here, and then the other expressing the donor, a normal functioning OTC gene. 7 subjects have been enrolled in this study, and the results, an update on the results will be provided on the 13th at the American Society of Gene Therapy meeting. But the first subject was enrolled at GOSH, and this was reported in Science, and more specifically in a UK publication on the outcome of that initial boy. Boy was born with OTC, neonatal onset OTC, had life-threatening hyperammonemic crisis at birth, and then enrolled in the summer of 2025 in the trial, the first subject. His name is Thomas, and was reported in a publication called The Independent. And 6 months later, was off all treatment, off all protein restriction, and essentially biochemically and clinically has been cured, and I understand continues to do well. So I just want to end with one platform that we are discussing with MHRA that we hope to, and also with HHS in the U.S., who's likely going to provide funding for this. And this is the idea of a mutation-specific approach, where you actually fix a mutation. One way to do that is something called a base editor. where you can convert a nucleotide mutation from a G to an A or a C to a T. This base editor technology has what, in this purple sort of blob, the base editor protein, and then there's a guide RNA. And the idea is, theoretically, that all you need to do is change the guide, because the guide is designed to target different mutations. So if this patient has a mutation in a particular gene, you design a guide for that. This patient has another mutation, you just design a guide, a different guide, and then finally that. And this was, the potential of this was actually surfaced in work done at my home institution, University of Pennsylvania, where they treated a newborn with a base editor, and the baby's name was baby KJ, and it really gained a lot of interest because at least it shows that it was feasible. However, the key is, how do you scale this? And that's what we've been thinking about and working with stakeholders here in the UK, specifically around this approach. Because while you can design guides specific to different mutations, that the base editor itself also has to be modified. So we heard from Reed early about the use of AI, and I never thought I would be there, but we actually are going to rely on AI to solve this problem. So we're working with a company called Profluent that is a protein engineer, has generated a CRISPR-like molecules based on protein design that gives you much greater flexibility. So the goal is there's a limited number of these editors that would cover the full spectrum of mutations, so that all that you really need is a guide for each mutation. So I'll just end with a description of this program and where we are. We're going to start with focusing on mutations that are more prevalent in rare disease populations. And the problem is recessive diseases are caused by many different pathogenic variants, but there are regional specific differences in the prevalence of these diseases. So we will develop a portfolio of base editors specific to a region that could be deployed when babies are born with those mutations. And this is where the work that you're doing here in Genomics England is going to be really useful. So we're partnering with them to identify the most common liver metabolic diseases and the most common mutations in each disease, and then develop base editors for these mutations, and working with MHRA to consider developing these as a basket. And I'll just end on a very positive note that we heard about the incredible leadership role that the UK is forwarding with Lawrence and his team at MHRA, that I think really will pave the way for the regulatory science to support this more bespoke approach. So with that, I want to thank you again for inviting us and for the work that you're all doing here, which is just inspirational for us. And you're going to probably be seeing a lot more of us in the UK. So thank you.
Thanks, Jim. That was fantastic. We have time for one or two quick questions before we break for lunch.
In the back? Yeah.
Hi. How are you, in your conversations with the MHRA, how are they treating different base editors? Will they be under one umbrella, under one platform? Are they seeing them as 3 different platforms? How are they seeing them?
I think that's to be determined. But in defense of health authorities, we as scientists have to bring data forward. We can't dictate sort of what we think is most convenient. I know you're not asking me that, but I do think that that the science will inform how to package those. The current thinking, from what I hear, is around a disease. So then you have different mutations for each disease, and that would be a platform. I don't think that's the way it should happen. With the work that Profluent is doing, if we have a limited number of base editors, deamidase, the scaffold, and the PID, then maybe the platform could be around the base editor that would provide more commonality between the different products. So I think this is a fascinating emerging new paradigm that the health authorities are there willing to work with us, but we've got to provide them data that would support how you could bundle these in a way in which you could then leverage information across those products from a safety and an efficacy standpoint. But from what I can tell, they're not only talking the talk, they're walking the walk in terms of working with us as we're thinking through this, which is remarkable. Coming from a country where things are not quite as organized and engaged as the situation with FDA right now.
I wondered whether you could comment on how we might achieve really, truly broad distribution across the brain, and whether that's going to need shuttles or other approaches. I just wondered what your thoughts on that are.
Yeah, I mean, I think the only way we're going to do it is penetrating the blood-brain barrier, and that there's been a lot of excitement about that. Probably 8 companies working on it, and unfortunately, the first application of that in humans went horribly wrong, and a four-year-old died of massive cerebral edema. We're gonna learn, I understand, more about that at the American Society of Gene Therapy, and I suspect, I have some idea what you're going to hear about, that would be very helpful to inform how to go back to whatever your technologies are to try to avoid that. You know, this whole business of bench to bedside and back, it's the bedside to back, but I still think that area is we've got to deal with some potential safety issues and how you model it, and really a very unfortunate way that field got launched.
Brilliant. Thank you, Jim. Thank you all for your great question and your attendance to this track. The track is resuming at 1:30, so I hope to see all of you back in the room for a very exciting second half of the day. Thank you.